field. So then,
Great. Well, just thanks so much, Raphael and Vince, for having me here. I'm really excited to be talking with you and to be sharing this session with a number of people whose work have really influenced the way I think about brain dynamics. And I think this is a really promising research area for scientists interested in understanding large-scale brain dynamics and their relationship to behavior. And there are many, many more controversies than we've talked about today or that I can cover in this talk. As such, I just want to bring your attention to our paper that discusses these issues in more detail. If you go up, it's on SciArchive. It's hopefully out soon somewhere. But this paper actually began at OHBM in Vancouver 2 years ago, where there's an open discussion. It's kind of evolved into what I think is a really nice review of the issues that we've been talking about today. And I want to emphasize that I'm the one standing up here, but this was really a team effort by this really amazing set of co-authors. And everything that I talk about today will be covered in much more detail in the paper. So if there are questions you have about references or things like that, feel free to ask me, but there's going to be a lot more detail in there. So I think let's just get started with like an idea of what we're going to be talking about today. So my talk will be in two parts. In part one, I'll review the diversity of methods for studying fMRI time-brain connectivity, and I'll guide you through the landscape of models and analysis approaches and how they relate to each other, discuss how different approaches provide different complementary insights into brain dynamics, and give a few ideas of things to consider when deciding on a TVC analysis strategy. In part two, I'll dive into a few of the controversies and open questions in the field, which I think are most salient and interesting. So let's get started. If you're going to only remember one idea from the first part of this talk, I think it should be that there's no single best method for studying time-varying connectivity, and that different modeling and analysis approaches provide complementary insights into brain dynamics. To get a sense of what I mean here, I think it's useful to take a few minutes to explore the landscape of ways which one might study time-varying functional connectivity. And this will allow us to get a sense of the high-level overview of how different approaches to studying time-varying connectivity relate to each other. So, because we're talking here about resting state fMRI, we're going to start with the bold fMRI time series, but a lot of these methods could equally be applied to electrophysiology data, or EEG, or even calcium imaging. And so from these bold time series, there are a few different analysis endpoints we might be interested in. So we might be interested in studying the time-varying properties of a particular connection, perhaps between two regions of interest, selected based on a specific hypothesis that we have about brain function or cognition. We could test the observed fluctuations against various null models. We could calculate summary measures like the mean and variance of the time-varying connectivity, or attempt to relate the fluctuations in connectivity to some other simultaneously recorded measures such as behavioral performance or measures of peripheral physiology. Moving beyond single connections, we might be interested in how patterns of whole brain connectivity change over time, and in measures from network science which summarize these patterns and the reconfigurations. If we take a state-based perspective on brain dynamics, we may be interested in measures like switching rate, state occupancy, or dwell times. And as with this case of single connections, we can compute brain state summary measures and compare them to null models to assess their time-varying properties. Now finally, as Michael just reviewed in the previous talk, we might want to try and actually model and understand the fast time-scale nonlinear neural dynamics that underlie the brain activity that we're able to measure with fMRI. And so at this point, we have a starting point, the observed bold time series, and a number of possible analysis and modeling endpoints. And so the question is, how do we get from one to the other? And so we heard early on in the beginning from Julia, sliding window is most likely the most common method for estimating time-varying connectivity. It's already being discussed in a little bit more detail, but as a review, this method involves just estimating time-varying connectivity by computing the correlation between two time series over successive windows. And once we have our estimates, we can calculate a number of summary measures. Now, importantly, however, the sliding window approach is by far not the only way to get to these metrics. So there are a wide range of other analytic approaches which also produce estimates of time-varying connectivity. And I don't have time to review them all here, and many of them have been mentioned already, but there are some examples like hidden Markov models and time frequency-based approaches. The critical point is that they all eventually produce estimates of time-varying connectivity that are in many ways functionally equivalent to those that result from sliding window correlation. That said, each method has its own particular assumptions, strengths, and weaknesses, and this is an important point that I'm going to come back to in a little bit. So moving from the case of individual connections is simply a matter of calculating time-varying connectivity between all pairs of time series. And again, this can be done using a variety of methods. And once we've estimated whole brain for time-varying connectivity, we can begin to explore the time-varying network properties. And now these network properties can also be applied to brain states, which, like TVC, can be estimated in multiple ways. So some methods, such as hidden Markov models, estimate states directly from the observed bold time series, where other common approaches involve clustering whole brain time-varying connectivity patterns, such as those calculated via sliding window correlation. And in either case, once we have our brain states, we can explore their time-varying properties. And then last but not least, studies of brain dynamics using biophysical models typically involve fitting the model to real brain data in order to set model parameters, which can then be interrogated for insights into the mechanisms underlying observed brain activity. And while this particular schematic depicts biophysical models as their own workflow, it's increasingly common to see modeling integrated into analysis of real brain data, and the use of these approaches in combination can provide powerful insights. So as you can see, the landscape of possible analytic and modeling approaches for studying time-varying connectivity is relatively large and complex, with multiple possible routes to the same analytic endpoint. And while it's generally difficult to cleanly categorize these approaches, it can be useful to make a basic distinction between data-driven approaches, such as those on the right, which analyze the properties of the observed bold time series without explicitly modeling the underlying neural dynamics, and biophysical models on the left, which focus specifically on these dynamics. Another way to distinguish between these methods is to break them down by what kind of insights they can provide us about brain dynamics. And so, going back to this point, that different approaches provide different insights. So, closest to the raw data are the estimates of time-varying connectivity. So these allow us to explore the relationship between momentary functional connectivity patterns and other time-varying phenomena like cognition or behavior. Models of brain states and their transitions allow us to investigate network properties of transient brain states and how they reconfigure over time. Comparing observed TVC estimates or summary measures derived from TVC estimates or brain state models allow us to test specific hypotheses about the temporal properties of neural time series. And then finally, biophysical modeling allows us to test mechanistic hypotheses about the phenomena that give rise to TVC and to explore the complex neural dynamics that may underlie the observed brain activity that we see with fMRI. And so given this diversity of methods, it can be difficult to decide which approach to use, even for those of us who are really familiar with these methods. So here are just a couple of things that I think are useful to consider when deciding on a TVC analysis. So there may be no single best method for studying TVC, and in addition to the fact that different methods can provide different and complementary insights into brain dynamics, each method also has its own set of strengths and weaknesses and assumptions. As such, the choice of what TVC method to use in a given situation often involves balancing trade-offs, and your choices should ideally be informed by your specific questions, your hypotheses and goals of your study, and the available data. So in terms of model assumptions, a good example is to consider the sliding window correlation approach versus hidden Markov models. So sliding window methods make a number of assumptions about the statistical properties of the time series data, but no assumptions about the temporal structure of brain dynamics. Hidden Markov models, on the other hand, specifically assume the presence of latent brain states when estimating time-varying connectivity, and that transitions between these states follow particular rules. Estimating brain states by clustering sliding window connectivity matrices doesn't involve making the same assumptions. Now, none of these approaches is necessarily better or worse than the others, but their differences can potentially impact your results, and you should keep this in mind when deciding which to use. So just to kind of review what we've talked about so far, there's a really rich landscape of methods for studying time-varying connectivity, and different methods provide different complementary insights into brain dynamics, and that when you're deciding on which method to use, you should think really carefully about what theoretical question you're trying to answer, because which method you use will determine whether you're actually able to answer the question that you're trying to get at, or will be trying to kind of force a square peg into a round hole. So in part 2, let's take a few examples of the controversies and outstanding questions in the study of resting time-varying connectivity. So first, I'll discuss the impact of arousal on brain function and behavior, and try to make the case that fluctuations in arousal should not be considered a confound. Second, I'll present evidence that both for and against the hypothesis that resting time-varying connectivity is related to ongoing cognition. And third, I'll detail how differences in data cleaning can impact time-varying connectivity analysis. And then I'll finally take a few minutes to talk about one way to think about the relationship between static time-varying connectivity and static connectivity and time-varying connectivity. So as we've heard from Shella, fluctuations in arousal can have a major influence on brain function and the measures of brain function. So as neuroscientists, we're ultimately interested in understanding what processes give rise to the varied neural phenomena we study. And so resting bold time-varying connectivity is certainly the result of a number of different underlying processes, ranging from intrinsic homeostatic processes all the way to brain activity supporting complex cognitive operations. And whether or not any one of these factors contributing to time-varying connectivity is considered a signal or noise really depends on the specific question being asked. And like Shell, I think that the global neural signals like arousal are a great example of this, especially in light of questions about the extent to which bold time-varying connectivity at rest is related to ongoing cognition. So this is a figure from a paper from Katie Chang from a few years ago, and what it shows is a simultaneously recorded fMRI, local field potential, and a behavioral index of arousal. And you can see that the behavioral index of arousal tracks the fMRI arousal index really well. And when you look at the percent of the variance that you can explain in the bold time series by this behavioral measure of arousal, depending on the region, you can explain up to 25% of the variance. So arousal is this global neural phenomenon that is really driving a lot of the change in what's happening in the brain over time. And I think kind of importantly, these fluctuations and durations in arousal are not just, they're not neural and non-relevant to cognition. They also are really important in cognition and behavior. So this is another study from the same year in which the authors looked at dynamic connectivity states that were associated with either an eyes-open brain state, which they considered a high arousal state, or an eyes-closed brain state, which they considered a low arousal state. And they measured this by looking at how open their eyes were. And so low eye scores are those in which the eyes are almost closed, and high eye scores are times when the eyes are wide open. And as you can see, in the eyes open brain state, you're more likely to spend time in this eyes open brain state when your eyes are fully open than when you're in the eyes closed brain state, and vice versa. But what I think is most interesting about this is that if you look at the probability of each state occurring during an auditory vigilance task, the number of times that you lapse in this task in which you have errors is correlated with the proportion of time that you spend in this low arousal brain state. And it's negatively correlated with the amount of time that you spend in this high arousal brain state. And so I think this kind of underscores a lot of work that has been done in the past, showing that arousal and these other global neural processes are really kind of fundamental to driving both behavioral variability and changes in cognitive function. Now, if we think about this for a second, it actually makes a lot of sense. So level of arousal, as Stella said, is largely driven by ascending brainstem neuromodulatory systems, which have widespread projections throughout the brain. Local and global release of neuromodulators has been repeatedly shown to have major impact on the dynamics of neural circuits, and it's been suggested that activity in these systems may be responsible for driving reconfiguration of functional brain networks. Given all this, it would be surprising if fluctuations in arousal were not closely related to time-varying aspects of brain function. From this perspective, far from being a confounding factor, fluctuations in arousal are an integral part of the phenomena of the systems and cognitive neuroscientists seek to explain. So the takeaway from this is that fluctuations in arousal have a major impact on brain function and cognition. And I think a lot of people, certainly in cognitive neuroscience, are really interested in time-varying connectivity at rest because it might offer an opportunity to study spontaneous and kind of self-directed cognition in a way that is not so easily accessible by other methods. And that we should think really carefully about whether these arousal and these global neural phenomena, whether someone's sleepy, whether someone's dozing off in a scanner, are really confounds. They may not be directly related to your question about working memory or perceptual vigilance or emotion processing, but they are really contributing a lot to the processes that are supporting the cognition that you're interested in. And so, speaking about this desire to try and look at how we can potentially use time-varying connectivity to study these spontaneous cognitions, like mind-wandering and daydreaming during the resting states. This is just one example of a recent study by Aaron Kucci, one of our co-authors, that looked at the connectivity between the posterior cingulate cortex and the medial temporal lobe subsystem of the default mode network. And what Aaron did is he had people during a painful stimuli paradigm in which they were experiencing painful stimuli. He asked them repeatedly throughout this experiment how often they were paying attention to the pain versus something else. So every couple of minutes he would ask them, what are you paying attention to? And this is called experience sampling or thought probe methodology. And when he looked at the static connectivity between these areas, he was unable to find any relationship between the amount of time that you were spent thinking about this painful stimulus versus thinking about other things. But if you look at the variability of time-varying connectivity, you can see that this does have, you do see a relationship, which goes back to a point made earlier, which is that for certain types of cognitive processes that fluctuate over short time scales, it may be that time-varying connectivity is more sensitive a measure than static connectivity. And so that provides a little bit of evidence that perhaps time-varying connectivity, even at rest, when we're not showing overt behavioral changes, is sensitive to the dynamics of cognition. But counter to that is the fact that, as Cheryl was talking about, many of the studies of the neural basis of time-varying connectivity were done in anesthetized animals. And so this is a figure from one of the papers from her group, which shows simultaneously recorded electrophysiology and bold fMRI, where you have the sliding window correlation on the y-axis, and over time you can see that, yes, you get time-varying connectivity both in these electrophysiology frequency bands and in the bold signal. So this is a little bit concerning. If we're interested in trying to use time-varying connectivity to study cognition, then a state like unconsciousness in which we wouldn't expect cognition to be occurring shouldn't show these types of fluctuations. And I think there's a little bit of nuance to it, because if you look at the dynamics and the properties of the time-varying connectivity, though it still exists, it does change. And this is actually one of the earlier papers on showing time-varying connectivity in humans and macaques that shows that the number of unique states that you visit and the number of transitions that you have between states decreases as you deepen, as your level of anesthesia deepens. And so while time-varying connectivity does remain during anesthesia, something about the properties of these phenomena is changing. And trying to pull apart what exactly the properties are that are sensitive to the things that you're interested in, whether it be cognition or behavior or clinical aspects, and which are related to these other factors which are contributing to the signal, is something that as a community we really need to work on, because as Phil said, there are so many things that contribute to it. And if you're trying to make a claim about the necessity of a relationship between a time-varying connectivity measure and your other measure of interest, it's important to make sure that it's not being driven by something else. And in terms of being driven by other factors, something that if you work with functional MRI, you're all too familiar with is this idea that we need to really denoise our data. Functional MRI, the signals that we're looking at are often, single-digit % signal changes, and in fMRI functional connectivity in particular, motion is a really pernicious problem. And so there's been a lot of talk in functional connectivity literature over the years about the impact of motion on estimates of functional connectivity, and that holds true for time-varying connectivity as well. There's been a number of papers in recent years looking at evaluating a very wide range of pipelines for what is most effective at removing the effect of noise on static functional connectivity, And this is the recent paper doing the same thing for one aspect of time-varying connectivity, which is the flexibility of a region's affiliation between different functional networks over the course of a scan. And as you can see, the different denoising pipelines have very different efficacy in terms of removing the residual effects of motion, with some pipelines showing essentially no residual effects of motion, and some pipelines like ICA or Aroma, which for static functional connectivity is generally believed to be one of the most effective pipelines at removing residual motion effects, not being quite as effective. And so motion isn't the only thing that comes in when we're doing denoising and data cleaning. Temporal filtering is something that we talked about at the beginning of the session in terms of what filtering should you be doing and how should that filtering influence your choice of window length if you're doing a sliding window correlation. So this is an example from a project that I'm working on and collaborating with people at Berkeley. And so what we're doing is we're applying a hidden Markov model which essentially tries to find brain states in resting fMRI data and looks at the probability at each time point that a state is active. And so because we wanted to make sure that our results were robust, we did this analysis through two different pipelines. But crucially, the pipelines were almost identical except for their frequency bands. Every other aspect of the preprocessing in terms of what our nuisance regressors were, in terms of like whether we included global signal or not, was identical. And so if we look at this state time series in which on the x-axis we have time and the y-axis we have states, you can see that the yellow is times at which that state is most likely to be active. This is through one pipeline with a temporal bandpass filter of 0.008 to 0.09 hertz. And if we look at this other pipeline, which is identical except for a couple of tenths or hundredths of a hertz difference, you show very different dynamics. And we think that this has to do not just with the small differences in the bandpass frequencies, but in the way that the filter is actually implemented. And one is using a discrete cosine filter, one is using a Butterworth filter. We're still trying to trace this up, but it suggests that when you're dealing with these dynamic phenomena, very small differences in how you're treating your data can make a big difference on the back end. And so that's just kind of one example of how pre-processing decisions can have a major impact on the results of TVC analyses. And while we have come a long way in the static connectivity literature in terms of figuring out what are best practices, what kind of denoising do you need to do for motion and other types of signals, it's still early days for time-varying functional connectivity. And there have only been a couple of papers that have looked at this really systematically. And until we are able to ensure that our results are not driven by differences in denoising strategies, it's going to be hard to come to a consensus on the extent to which time-varying connectivity is associated with clinical differences or cognition. The final controversy that I want to talk about today is one that's a little bit more conceptual, and it has to do with the fact that static functional connectivity networks are really stable. And this has been a really good thing for a long time. One of the benefits of resting state functional connectivity is that you can scan someone a year apart, and their connectome will be so similar that you can identify them in another group of people. And that's great. It means that it's a stable biomarker measure. It can be used to perhaps distinguish people based on disease state. But at the same time, it suggests that the resting state functional connectivity is maybe not so sensitive to the kinds of changes in cognition that many people are interested in. So this is a figure from a paper from Michael Cole from a couple of years ago showing the functional connectivity patterns from a number of different tasks. And showing their similarity both to each other and to the resting state. And the correlations are really high. And this and other papers have estimated the correlation between your patterns of functional connectivity at rest and during task execution can be up to something like 0.9. And this is pretty well replicated across studies. And so then the question is, if we're seeing such stable patterns of static functional connectivity, even when we are doing overtly different states, when we're doing a relational task versus a gambling task, something that is really kind of pushing us to use different types of thoughts, why should we expect to see changes in time-varying connectivity structure during a period of rest in which you're just sitting in the scanner, you're not being told to do different things, and maybe the only difference is that you're kind of mind-wandering about different things in different ways. And I think the resolution to this has to do with something that came up earlier, which is the idea that static functional connectivity is a summary measure, right? And like any summary measure, it's an aggregate measure. This aggregate measure of static functional connectivity is likely going to fail to capture some aspect of the underlying dynamic phenomena. An example of this is that the same pattern of static functional connectivity might result from different spatial-temporal patterns of underlying time-varying connectivity. And I think this has to do with the fact that brain dynamics and behavior unfold over time in a particular sequence, right? The words that I'm saying to you would mean something very different if I said them backwards. And in the same way, the trajectories which my neural systems take through time are meaningful, and that if we permuted them, they're not time invariant. And so this means that even if you have the same distribution of static connectivity, of connectivity states, they can have very different temporal profiles. So an example might be if we have two brain states, brain state A and brain state B, and they each show up 4 times. Well, you could have them show up like this, in which there's kind of a block structure, or they could show up interspersed, or they could show up in a kind of a mini block structure. And you can think about this as an example of, instead of 4 states, maybe you have 180 volumes, or you have, an hour of resting state data. And if you average over this time, you may see, well, there's an equal number of A's and B's in each case, but the dynamics and the temporal sequence in which they occur is different. And that's going to be meaningful if we think that the mental capacities come from brain activity. And so I think that it's important to go beyond just identifying functional connectivity patterns at a high temporal resolution. I think that there's a lot of utility in trying to see what are the momentary functional connectivity patterns associated with, specific cognitive processes, like what is the delay-related functional connectivity pattern during a working memory experiment. But to really understand the brain dynamics and how they give rise to cognition and behavior, we need to understand how they unfold over time. And so I think this is just kind of one way of saying that resting state time-varying connectivity is consistent with stability of static functional connectivity, and that the temporal structure of time-varying connectivity fluctuations may be key to understanding brain dynamics and how they relate to cognition and behavior. So just to kind of wrap up quickly and talk a little bit of like what we've learned and what we don't know yet, there's a really rich landscape of methods for time-varying connectivity, and there's no one best method. And they provide complementary insights into brain dynamics. If you're going to be choosing a time-varying connectivity analysis, I'd say work backwards from what your hypothesis is and how you think the brain works. If you think that the brain visits distinct states, then maybe go with a state-based approach. If you don't think it visits distinct states, maybe try another approach. And we don't know the ground truth answer to this. And so there's really utility in using both of these methods and looking at how they provide different insights and different evidence about the brain phenomena that we're interested in. We talked about how fluctuations in arousal are likely a major factor contributing to bold time-varying connectivity, and that arousal may drive behavioral variability and functional reconfiguration, and you should think really carefully if you consider arousal a confound. We talked about how there's emerging evidence that resting state time-varying connectivity is relevant cognitively and behaviorally, but that important questions remain about this relationship. It's clear that there are some aspects of time-varying connectivity that remain during states of unconsciousness in which we would expect no changes in cognitive state at all. We talked about how preprocessing and denoising can have a major impact on the results of time-varying connectivity analysis, and that we're really lacking consensus best practices for many of the components of the denoising pipelines that we need to use to make sure that our data is robust. And finally, we've talked about how stable patterns of static functional connectivity are conceptually consistent with time-varying connectivity that changes during rest. Just because static connectivity patterns are relatively stable between overtly different task states doesn't mean that their underlying temporal patterns are the same. And so with that, I just want to acknowledge mostly these wonderful co-authors who have worked with me for the past 2 years on this paper. I think they've put an immense amount of work into it. And in particular, I wanted to thank Dan Kessler, who wasn't able to be here, and Vince Calhoun, who were both kind of helping me organize this effort and wrangle this huge list of co-authors. I want to thank my lab at UC Berkeley, my funding, and just a plug for my poster, which has nothing to do with time-varying connectivity, but a lot to do with dynamics. And if you're interested in timescale stuff, you might want to come to that on Monday afternoon. And I'm happy to take questions.